In today's lecture, we will study histology of a muscle. So the cells of the muscular tissue are known as myocytes. Embryologically, muscles are derived from mesoderm. Muscle is one of the basic tissue of our body. We know the four basic tissues are epithelium, connective tissue, muscle and nervous tissue. So the muscles are contractile. They have contractile filaments which will help in the movement. Functions of muscle are they produce movement, they maintain posture, they stabilize joint and they release heat. So they help in thermogenesis. Functionally, the muscles are excitable, means they respond to chemicals released from nerve cells and they can generate signals over the membrane. Then they have contractility, they can shorten and generate force. They have extensibility, they can stretch without damaging, and they have elasticity, they can regain their original shape. Some terminologies related to the muscle are plasma membrane of the muscle is known as sarcolemma. Cytoplasm of muscle is known as sarcoplasm, and the smooth endoplasmic reticulum present in the muscle is known as sarcoplasmic reticulum. Mitochondria are known as sarcosomes. Histologically, the muscles are classified into two types. Those muscles which show presence of striations are known as striated muscles, and the skeletal muscle and cardiac muscle are striated muscles. And those muscles which under microscope do not show striations are known as non-striated muscles. Example is smooth muscle. Physiologically, muscles are classified as voluntary and involuntary. Voluntary muscles are those which function and their function is under control. So example is skeletal muscle, and those muscle whose functioning is not under their control is involuntary muscle. Example is cardiac and smooth muscles. Thus, there are three types of muscles: skeletal muscles, cardiac muscles and smooth muscle. First, we will see the histology of skeletal muscle. Skeletal muscles are known as striated muscles because they show striations. These are voluntary muscles because their action is under control or under our will. And as they are attached to the skeleton, they are also known as skeletal muscles, and they are mainly responsible for locomotion. So the upper limb and lower limb skeleton shows presence of skeletal muscle, but there are some viscera which show skeletal muscle. So microscopically these are identical to skeletal muscle. So such viscera are tongue, pharynx, peripheral part of the diaphragm and upper two-third of a esophagus. So they have, though these are viscera, but they have skeletal muscles in them, and these muscles are involved in speech, breathing, swallowing and coughing. In this slide, we will see the structural framework or supportive framework of the skeletal muscle. So when we take a transverse section of skeletal muscle, we can see that the entire skeletal muscle is covered by a connective tissue framework known as epimysium. So the epimysium covers the entire skeletal muscle. This skeletal muscle is formed by numerous bundles or fasciculi. So these are the bundles or fasciculi of the skeletal muscle. So this is one bundle, this is other bundle, and these bundles are covered by connective tissue known as perimysium. So this blue colored structure is the perimysium, which covers individual muscle fasciculus or muscle bundle. Now how this muscle bundle is formed? It is formed by numerous myofibrils. So these are all myofibrils which are again covered by the endomysium. So the blue covering over the small myofibrils is the endomysium. Thus the myofibrils are covered by endomysium, the bundle or fascicula of muscle is covered by perimysium, and the entire muscle is covered by epimysium. And the endomysium consists of basal lamella, reticular fibers and connective tissue. The same you can see in this diagram. So this is the entire muscle covered by epimysium. Then the muscle fascicles are covered by perimysium, and individual muscle fiber is covered by endomysium. And within this connective tissue framework, we have the blood vessels and nerves. When we see the slide of skeletal muscle under microscope, we can see that the skeletal muscle fibers are long, cylindrical, these are elongated and they are unbranched. Here, the nucleus is shifted towards the periphery. They are present just beneath the sarcolemma, and the skeletal muscle fibers are multinucleated. So it is forming a syncytium, and the nuclei is shifted towards the periphery by the myofilaments present in the sarcoplasm of skeletal muscle. Then the sarcoplasm will show alternate dark bands and light bands. So we can see cross striations in the skeletal muscle. The dark bands are known as A band and the light band is known as I band. A stands for anisotropic, means the light rays can't pass through it, and I stands for isotropic because the I band has the power to refract the light in polarized light microscopy. In this slide, we can see the ultrastructure of skeletal muscle. So this is the skeletal muscle. Entire muscle is covered by the epimysium under which lies the sarcolemma. We can see the peripherally placed nucleus also, and it is covered by now the cell membrane of the skeletal muscle is known as sarcolemma, and this muscle is formed by many muscle fasciculi. So these are the muscle bundles or fasciculi. These are covered by perimysium, and the fascicles are formed by numerous myofibrils. Each myofibril is covered by endomysium. So if we see the ultrastructure of this myofibril, it has the contractile myofilaments known as actin and myosin. So there are two types of contractile myofilament. So here the blue colored thin filaments are actin, and the thick red colored are the myosin filaments. So we will correlate this figure with this figure. Okay. So in the skeletal muscle, we see alternate cross striations. So there are alternate dark bands. So this much area is the dark band, and then we have the light band. So this is the light band. Now why these dark bands and light bands are produced, we'll see. So the blue colored is the thin filament, it is the actin filament. Okay, so this is the thin actin filament. So in the area of light band, we have only actin filament, the thin actin filament. That's why it is light band. And in the light band, there is a central dark band line. It is known as Z line. So Z line is a dark line present in the light band. Okay, and the light band is formed by actin filaments. Now coming to the dark band. So now this is the dark band. So if we see the periphery of the dark band, there is overlapping of actin filament with the thick myosin filament. So this zone is comparatively darker, whereas in its central part only myosin filament is there. So in the dark band, the peripheral area is more darkly stained because there is overlapping of actin and myosin filament, whereas in the central part there is comparatively lighter zone where only myosin filaments are there, and this zone is known as H zone. Again in the H zone we can see a central dark line which is known as M line. Okay, so there are alternate dark bands and light bands. In the dark band we have a light zone known as H zone. In the light H zone we have a dark line known as M line. So and in case of light band we have a central dark line which is known as Z line. Now the area between the two adjacent Z line, now this is the Z line of one light band and this is the Z line of other light band. So the area between the two adjacent Z line is known as sarcomere, and sarcomere is the structural and functional unit of a skeletal muscle. So now we in the ultrastructure we know that the myofibrils are the myofilament are the protein fibers, and these fibers are made up of repeating myofilaments, that is actin and myosin filament, and these repeating myofilaments form an area between the two adjacent Z line is known as sarcomere. Sarcomere has three types of proteins: structural proteins, contractile proteins, and regulatory proteins. So the actin filament or thin filaments are covered by troponin and tropomyosin, which are the regulatory proteins. Thus, in the ultrastructure of skeletal muscle, we can see the contractile myofilament, actin and myosin. These filaments will form alternate dark band, that is A band, and light band is I band. In the dark band, we have centrally placed lighter zone known as H zone. In the H zone, we have a central dark line known as M line. In the light I band, we have a centrally placed dark line known as Z line, and the area between two adjacent Z line is known as sarcomere, which is the structural and functional unit of skeletal muscle. In this diagram, we can see the two contractile proteins or myofilaments. So this is the actin filament and this is the myosin filament. So the actin filaments are thin filaments, and the actin filaments are governed by two regulatory proteins. So the first is troponin and second is tropomyosin. Okay. Now in the myosin filament, we have the head of myosin. The head of myosin has two sites of attachment. In the actin filament, there are two regulatory proteins, troponin and tropomyosin. And in the myosin filament, we have two sites of attachment over the head of myosin. One site is for actin and other site is for ATP. So these are helping in the contraction of the muscle. So how the muscle contracts? Calcium is released from the sarcoplasmic reticulum through T-tubule. It will pass into the sarcoplasm. The calcium will bind with troponin. So these are the troponin. Calcium will bind with troponin, and thus after binding there will be modulation in the tropomyosin, and these tropomyosins will move away. They will release and they will expose the site of attachment with the myosin. So the actin has a site for attachment with the myosin, and that is exposed when the tropomyosin is released. And then the myosin head has one site for attachment with the ATP. It will, sorry, one site of attachment with the actin, so it will bind with the actin. It will form actin myosin contraction, and the other site is for ATP. So once the ATP is attached to the myosin, the muscle contraction occurs by formation of actin myosin complex. Smooth endoplasmic reticulum in the skeletal muscle is known as sarcoplasmic reticulum. It is very important structure. It is well organized in skeletal muscle, and it is the reservoir of calcium. And the dilated part of the sarcoplasmic reticulum is known as terminal cistern. Now when you concentrate on the diagram, you can see numerous myofibrils. The myofibrils will unite to form the muscle bundle or fascicle, and such bundles will unite and they will form the muscle. So the muscle is covered by that green colored epimysium, and over there is sarcolemma, and we can see peripherally nucleated nucleus also. Now just below that we can see the blue colored is of sarcoplasmic reticulum, and its terminal dilated part which is accompanying the T tubule on either side is the sarcoplasmic cistern. It is known as terminal cistern, and in between the two terminal cistern, the green colored structure is T tubule or transverse tubule. So it is the invagination or ingrowth of sarcolemma, and in the skeletal muscle it is present at the junction of I band and A band, whereas in cardiac muscle it is present over the Z line, and the transverse tubule is transverse to the long axis, and thus the skeletal muscle forms a triad. So there are two terminal cisterns on either side of the T tubule, and their main function is to release calcium, and this calcium is transmitted to the sarcoplasm. At the neuromuscular junction or motor end plate side of the muscle, it is the junctional complex of axon terminal and muscle fibers. So neuromuscular junction is the junctional complex of axon terminal, which you can see in the diagram. The yellow colored axon terminal is there, and on other side we have the muscle fiber. So the terminal end of the axon is known as terminal button, and it is myelinated, and it shows presence of vesicles, secretory vesicles, which contain acetylcholine and number of mitochondria. So, and on the other hand we have the motor end plate or solo plate. So it forms number of foldings that contain receptors for acetylcholine. So this is the side of muscle where acetylcholine receptors are there, and in between the axon terminal and the motor end plate of a muscle there is synaptic cleft. It is a gap of about 40 to 100 nanometer between the axonal nerve terminal and the motor end plate of a muscle. In these slides, we will see how contraction and relaxation of the skeletal muscle occurs. So, the at one end we can see the axon, and the axon terminal has got synaptic vesicles which are filled with acetylcholine. Acetylcholine is released in the synaptic clefts and then to the motor end plate, and thus the muscle is stimulated by the nerve, and then calcium is released into the sarcoplasm through T tubules. Okay, and once the calcium is released it will bind with troponin C. So we know that two contractile filaments are actin and myosin. So actin has two structural proteins over it, troponin C and tropomyosin. So when the calcium binds with troponin C, it will modulate the tropomyosin, and thus the tropomyosin is released from the actin, and now the site at which the myosin will attach over actin is exposed, and at that site of exposure the myosin head will get attached. Again we know that the myosin head has got two sides, one side is for ATP and other side is for actin attachment. So the myosin binds with the sides of actin filament. After binding of myosin head with thin filament, there is attachment of ATP with myosin head, and this will cause contraction of the muscle. So we will revise it briefly again. After nerve stimulation, calcium is released from T tubules into the sarcoplasm. Calcium will bind with troponin C. Troponin C is one of the structural protein of actin filament. So once the troponin C binds with calcium, it will modulate the tropomyosin, and tropomyosin is released from the actin, and thus the site for attachment with the myosin over actin is exposed. So the myosin head will attach with the actin, it will form actin myosin complex, and the myosin has again other site for attachment of the ATP, and after that the muscle will contract. And as you can see in the diagram of contracted muscle, the H zone will disappear when the muscle is completely contracted, and when the muscle is relaxed, the H zone will expand. When the muscle relaxes, there is release of myosin head from the actin, and there is conversion of ATP to ADP. So the binding of myosin head will weak the binding of myosin head with new actin site, and there is release of inorganic phosphate, and it will release ADP. And after the power stroke of myosin head, again the binding of myosin head will occur with new actin filament. So this is the repetition of the cycle. So in short, what happens in relaxed state is the myosin head is released from the actin binding site, and there is conversion of ATP to ADP. And when the muscle is relaxed, the I band and Z zone are expanded. There are two types of fibers in skeletal muscle, red fibers and white fibers. So the red fibers have more amount of of hemoglobin, so they appear red in color, and the white fibers have more amount of glycogen. And the mode of energy generation in red fibers is through oxidative phosphorylation, and in white fibers it is by anaerobic glycolysis. Coming to the applied importance of skeletal muscle, so myasthenia gravis is an autoimmune disease in which autoantibodies are formed against acetylcholine receptors. So we know that the acetylcholine is an important neurotransmitter at neuromuscular junction, and if the autoantibodies are formed, they will damage the acetylcholine receptors, and the muscles will slowly become weak. Then the neurotoxins are secreted by snake. So the snake venoms are neurotoxic. They can again bind with the acetylcholine receptor and paralyze respiratory muscles and cause death of the individual. Then the third term is stretch reflex. It is myotactic reflex, and it is illustrated by a blow upon muscle tendon that in reflex induces muscle contraction. So most common stretch reflex we elicit is biceps reflex. As the name suggests, cardiac muscles are located in heart, and the peculiarity of cardiac muscle is that they show autorhythmicity, means they contract on their own without any stimulation, and hence it is also known as pacemaker. So the SA node, sinoatrial node, is known as pacemaker, which has autorhythmicity. Then the cytoplasm or sarcoplasm of cardiac muscle shows striations, but these are not so obviously visible like in skeletal muscle, and the nucleus is centrally placed. It is mainly supplied by autonomic nervous system, which includes sympathetic and parasympathetic component. So sympathetic will stimulate the cardiac muscle and they will increase the heart rate, whereas the parasympathetic component by vagus will inhibit the activity of heart muscles. Cardiac myocytes are connected with each other by intercalated disc, and they also show side branching. Then the cardiac muscles are thus striated involuntary muscles. They are striated because they show cross striations, and they are involuntary. The action of these muscles is not under voluntary control. Then the cardiac muscles show less number of myofibrils, and that's why the striations are less prominent. The cardiac muscle fibers accommodate more amount of mitochondria and sarcoplasm. When we see the cardiac muscle under microscope, the myocytes will appear long, cylindrical, thick, and they are showing branching fibers. So the muscle fibers of cardiac muscle shows branching pattern. Then the cardiac muscles are joined with one another by intercalated disc, which gives functional syncytium. So the junctional complex between the adjacent cardiac myocyte is known as intercalated disc. So here we can see the thin dark lines. These are intercalated disc. Also in the in the cardiac muscle we get striations, but if we compare it with the skeletal muscle, these are less stained. Okay, and each cardiac myocyte has a single centrally placed large oval nucleus, and these myocytes are enclosed in loose connective tissue in which the blood vessels and nerve fibers are present. Cardiac myocytes are of four types. First variety is nodal or pacemaker. These are present in SA node and AV node. So these type of cells, myocytes, are myogenic, and the parasympathetic innervation by vagus will diminish the heart rate. And the second variety is transitional cells. So these are present in the AV node and bundle of His. Third type of cardiac myocyte is Purkinje cells. So they are located in two fascicula of AV node, AV bundle, and the Purkinje cells also connect the SA node with AV node. And the Purkinje cells are large, pale, less striated, and the most important thing is they have high conduction velocity. The ordinary working myocytes and the nodal and transitional myocytes, they are highly excitable, but the conduction velocity is less. So maximum conduction velocity is present in Purkinje cell, but the Purkinje cells are less excitable, whereas the nodal cells, transitional cells, and ordinary working cells, they are highly excitable, but the conduction velocity is low in these cells. Intercalated disc is also known as line of Ebert. So normally a cardiac muscle is formed by muscle cells known as cardiomyocytes. These cardiomyocytes are connected with each other by a junctional complex known as intercalated disc. Intercalated disc helps the cardiomyocytes to function as a single unit, a single functional syncytium. It also helps in synchronized contraction of cardiac tissue. Under microscope, the intercalated disc appears as a thin dark line. It has got step ladder like appearance, and these intercalated discs are present between the adjacent cardiac muscle cells. So they are mainly forming functional syncytium, and there is a gap junction between the two cardiac muscles. Now if there is any mutation in the intercalated disc, it can lead to cardiomyopathies. And under microscope, if we see ruptured intercalated disc, it may be due to ventricular fibrillation or electrical injuries. In this slide, we can see the ultrastructure of cardiac muscle. So the cardiac muscle is formed by myofibrils, and in this well labeled diagram we can see the myofibrils are covered by sarcolemma. And if we go a bit below, we can see the blue structure is labeled as sarcoplasmic reticulum, and the green tubular structures are the T tubules. So in the cardiac muscle, the sarcoplasmic reticulum of the cardiac muscle is not well organized, and there is only one terminal system. If we compare it with the skeletal muscle, in the skeletal muscle, sarcoplasmic reticulum is well developed. We have central T tubule, on either side we have sarcoplasmic reticulum, which forms the sarcoplasmic cistern, and thus a triad is formed. But in cardiac muscle, the main difference is that the sarcoplasmic reticulum is not well developed. So we have only one terminal cistern accompanying the T tubule. So it forms a dyad instead of triad. So in cardiac muscle we have dyad in which a small cistern is there and T tubule is there. Again, other difference, if we compare it with the skeletal muscle, we know that the T tubules in the skeletal muscle are present at the junction of A band and I band, whereas in cardiac muscle, the T tubules are present at the level of Z lines. Smooth muscles are non-striated. They do not show cross striations. Though actin and myosin filaments are present in the smooth muscle, but they are not arranged in regular fashion. So we cannot see them under microscope. Then the smooth muscles are involuntary because their action is not under willpower or voluntary control. So the smooth muscles thus are non-striated and involuntary muscles. They are supplied by autonomic nerves that include sympathetic and parasympathetic component, and the smooth muscles are connected through gap junction, means there is a space of about 40 to 80 nanometer between adjacent smooth muscle cells, and here we get the gap junction, and it helps in simultaneous contraction of adjacent cells, that is myogenic activity is possible. And the smooth muscles do not have T tubules. Instead of T tubules, they have caveoles in them. Caveoles are the invagination of sarcolemmas, and these caveoles are surrounded by sarcoplasmic reticulum. Smooth muscle is located in gastrointestinal tract, blood vessel, respiratory tract. It is present in ureter, uterine tube, vas deferens, urinary tract, and erector pylorus. It is also present in ciliaris and sphincter pupillae muscle. When we see the muscles under microscope, the smooth muscle in the longitudinal section shows spindle shaped cells. So these are the spindle shaped cells, and these are filled with dark eosinophilic cytoplasm or sarcoplasm, and it is dark eosinophilic because it has myofilaments in it. So the myofilaments, that is actin and myosin, are present in all the three varieties of muscles. Now the nucleus is present in the center. It is flattened, elongated, and it is present in the center. Then if we see the transverse section of the smooth muscle, then these muscle are arranged in the form of bundles. So these are the muscle fascicular muscle bundle. So this is one muscle bundle, this is other muscle bundle, and these muscle bundles are formed by fibers which are known as myofibrils. So the transverse section of skeletal and smooth muscle appear somewhat same, only the difference is that in skeletal muscle the nucleus is peripherally placed, whereas in smooth muscle the nucleus is present in the central part. Coming to the ultrastructure of smooth muscle. So this is the spindle shaped smooth muscle cell. So this is individual cell of the smooth muscle, and the space in between the two adjacent smooth muscle cell is about 40 to 80 nanometer. So there is a gap of about 40 to 80 nanometer between the adjacent cells, and this gap is filled by elastic fibers, collagen fibers, and reticular fibers that surround the muscle. And if we see the composition of this spindle shaped fiber, it is first covered by sarcolemma. So the outer membrane is the sarcolemma. And now coming to the sarcoplasm. So the sarcoplasm has fibrillar and non-fibrillar component. Non-fibrillar includes all the cell organelles like nucleus, mitochondria, Golgi apparatus is there. Then we have ribosomes, then endoplasmic reticulum. They are all non-fibrillary. And in the fibrillary component, mainly we have cytoskeleton and contractile filament. So we can see this is the cytoskeleton and contractile filaments. Contractile filaments are the same, actin and myosin. So here we can see it is forming a spindle shape. Now these thin filaments are actin, and you can see the central placed is thick filament or myosin. So here we can see actin myosin complex formation. So this is the cytoskeleton. Cytoskeleton is mainly formed by some proteins like desmin, vimentin, hamin, and the actin is also important because it forms internal supportive framework in the cytoplasm or sarcoplasm. Means actin is responsible for maintaining the spindle shape of the smooth muscle cell. And the contractile filaments are thin filament or actin and thick filament or myosin, and both will form actin myosin complex. Now again, one new terminology here is caveoli. So these caveoles are invaginations from the sarcoplasm which communicate with outside. And if we compare the smooth muscle with the striated or skeletal muscle, then caveoles are compared with the T tubules, and that's why these are in close relation with the sarcoplasmic reticulum. So once the calcium is released, it will pass from sarcolemma to sarcoplasm through these caveoles. And again, other important term is dense bodies. So these are the dense bodies. We can see spindle shaped contractile filaments are there, mainly the actin filaments. So these all thin actin filaments, these are attached to the dense body, and dense bodies are compared with the Z lines of the skeletal muscle. How does the smooth muscle contract? So first there is depolarization of sarcolemma, from there to the caveoli, and then to the sarcoplasmic cistern. So the sarcoplasmic cistern will release calcium ions and ATPase. Calcium will bind with the calmodulin, and the actin will slide over the myosin, and thus the muscle will contract. So peristalsis is the wave of constriction. It is caused by contraction of circular smooth muscles. Depending on nerve innervation, there are two ways of stimulating a smooth muscle. So first is unitary smooth muscle and other variety is multi-unit smooth muscle. So the basic difference between these two varieties that in unitary smooth muscle, a single nerve fiber will supply a single cell, and then the nerve impulse will transmit from one cell to other cell via gap junction. Whereas in multi-unit smooth muscle, each and every cell of the muscle has got individual innervation. So the unitary smooth muscles have myogenic contraction, means a single nerve impulse will be transmitted from one cell to other via gap junction. And the example of unitary smooth muscle is walls of gastrointestinal tract. We have seen the movement of peristalsis where the contents are propelled forward. So it is an example of unitary smooth muscle. Then uterus, uterine tube, urinary bladder and ureter also show myogenic activity. And the multi-unit smooth muscles are those in which a single cell is supplied by its own nerve fiber. So all the cells have their own nerve fibers. So the action of such muscle is quicker and faster. An example is iris and vas deferens. Apart from muscle, there are some other contractile cells present in the body. So they are myoepithelial cells, myofibroblast, myoid and myointimal cells.